Good afternoon, dear viewers. I am Dr. Amit Bhattacharya, Associate Professor in Department of Zoology, Ramjas College, University of Delhi. Today, in this part of the lecture, which is a part of the biochemistry series, I will be talking about the understanding of the enzymes, the structure, classification and the function. So, this is the second part of this lecture in which I will be talking about the classification and the functions of the enzyme. Now, before going into the functions and the enzyme kinetics, we should know that how exactly the enzymes are being classified. So, the enzymes are basically classified depending upon the reaction which the enzyme are doing, what is the substrate specificity which they have. So, the enzymes have been classified systematically into various classes. So, there are 6 major classes of enzymes which have been classified, and there is an enzyme commission which is there. Now, this enzyme commission is the body which is responsible for giving up a nomenclature to each of these enzymes so that it becomes easy for any of the scientist or the working researcher to classify what exactly that enzyme is. So, this is called as the enzyme code. So, each of the enzyme has been given an enzyme code which is shown over here. So, it shows EC, then there is a major class 1, 2, 3, 4, 5, 6, then there are 2X which shows the various subclasses of that particular major class. Now, the first number of the enzyme commission, it classifies the enzyme reaction into 6 major groups. So, the first one is the oxidation reduction or the reductase. Then comes the transferases, hydrolases, lyase, isomerases and ligases. Now, this is a tabular representation which shows the various 6 major classes over there. So, if we see over here, the example which is shown is the lactate dehydrogenase. So, EC is the enzyme commission. So, 1 shows the major class, then there is 1.1.1.27 which shows the other subclasses under that particular major class. So, the class are divided into 6 major categories, the major classes. The first one is called as the oxidoreductase, which are majorly responsible for oxidation and reduction processes. The second one is called as the transferases, which are responsible for the group transfer. So, wherever there is a group transfer, the transferase enzymes are responsible. Then comes the hydrolases, which brings about the hydrolysis reactions over there. Then the lyases, which adds or removes the functional group to form the double bond over there. This fifth one comes is the isomerases, which are basically responsible to forms the isomer. So, one of the example is the triose phosphate isomerase, which is present in the glycolysis reaction, and this final one or the sixth one is the ligases, which are responsible for ligation of the 2 substrates at the expense of the energy or the ATP molecule over there. So, we will see each of them with the help of one example. The first one is called as the oxidoreductase. So, as the name is suggesting, there is an oxidation reduction which this particular enzyme is bringing about. So, if we see over here, what is happening is the lactate in the presence of NAD+, so the NAD+ gets reduced to form the NADH, and in this reduction process, the lactate releases its proton, which is taken up by the NAD+ to form NADH, and finally the pyruvate molecule is formed over there. So, this is the oxidoreductase type of reaction which is shown over here. The next one is called as the transferases. So, many of the transferases we come across during the protein metabolism, especially during the transamination reactions over there. So, over here the example which is shown is called as the alanine aminotransferases, which transfers the alpha amino group between the alanine and the aspartate. So, over here if we see from the how the amino group is moving from one molecule to the other molecule, forming the other thing. So, there is a transfer of the reaction which is taking place, and this particular reaction is called as the transferases. Now, the next third part of the category is called as the hydrolases. So, if we see over here, there is an enzymatic reaction in which the adenine and the base are being separated in the presence of the water molecule. So, what is happening is there is a hydrolysis reaction which is occurring over there, and the enzyme is called as the hydrolases. The fourth enzyme classification is the lyases, in which the water is added to reduce the double bond over there. So if we see over here, this is an example which is shown from one of the step within the citric acid cycle, where the fumarate, where there is a double bond, it gets reduced to a single bond by addition of a hydroxyl group given up by the water molecule to form the malate over here. So this is a lyase kind of example where the enzyme is called as the fumarase over there. Now the next enzyme classification is called as the isomerases. So the example which is shown over here is an example from the glycolysis pathway. The dihydroxyacetone phosphate, which is an isomer of the glyceraldehyde 3-phosphate. So in the presence of the enzyme triose phosphate isomerase, what is happening is the dihydroxyacetone phosphate is forming the glyceraldehyde 3-phosphate. So the isomers are formed. So one is a aldehyde, one is a ketone form of that particular molecule. The next one is called as the ligases. So as the name is suggesting, there is a ligation reaction which is occurring over here. So the tRNA to the tRNA, there is an addition of the amino acid. So this is an example from the protein translation process. So we can see over here, there is a high energy bond which is formed over here, the tRNA and the incoming amino acid, and there is a linkage of the amino acid to the tRNA is occurring with the help of the ligases molecule over there. So these are the 6 major classes of the enzyme which have been classified based upon what kind of reaction they are undertaking and what is the specificity of the substrate to which they are acting on. Now the enzyme kinetics was best described in a reaction or the equation which is called as the Michaelis-Menten kinetics. Now these are the two scientists who came up with the Michaelis-Menten kinetics. So what they said that there is a variation in the, whenever there is a variation in the substrate concentration, the reaction or the catalysis also varies up. So each of the enzyme is having a rate of catalytic reaction which is called as a V0, and when it reaches the maximum velocity of its catalysis, it is called as the Vmax over there. So they said that the rate of catalysis reaction, it rises linearly as the substrate concentration increases, but after a certain point all the enzyme has been attached with the substrate. So there is an enzyme substrate complex, so there is a saturation level which has occurred over there, and finally when it reaches the upper plateau where there is a maximum higher concentration of the substrate concentration, but there is no free enzyme, so it reaches the maximum velocity which is called as the Vmax. So this particular enzymatic kinetic reaction, it discusses about the rate of catalysis which rises linearly as the substrate concentration increases, and then it begins to lay off at a particular point, and finally it approaches a maximum point where there is a high concentration of substrate and there is no free enzyme because all the enzymes have been attached or engaged with the substrate over there. Now if we see the plot over there, this is called as the Michaelis-Menten plot, which discusses about this particular enzymatic reaction. Now as I told you, as the reaction initiates, there is a linear increase of the catalytic rate of reaction, so the reaction velocity increases as the substrate reaction increases, and finally it reaches a phase which is a layoff phase where there is all these enzymes have been bound to the substrate, so there is a saturation which has occurred, and finally it reaches a plateau where there is a higher concentration of substrate, but there is no enzyme which is free to bind to this substrate, so it reaches the maximum velocity which is called as the Vmax. So the Michaelis-Menten plot came up with a reaction or the equation which is called as the V0 is equal to Vmax S, which is the substrate concentration, divided by the Km. Km is the Michaelis-Menten constant which is there, and S is the substrate. So this describes the initial velocity. When we know the initial velocity, we know the maximum velocity or the substrate concentration, we can plot that one, and then we can take out what will be the maximum velocity of this particular enzymatic reaction over there. So this plays a very important role in understanding all the equations and the functioning of the enzymes, because if we see any of the books, apart from the enzymes name, there will be enzyme kinetics which will be also mentioned about that particular enzyme. So knowing that particular thing is also important for understanding how exactly the enzyme functions, what are the specific substrate to which the enzyme prefers and binds to, and how the reaction process goes about. Now, if we see over here, the Michaelis-Menten constant is called as the Km. So, we just discussed the V naught is equal to V max S divided by Km plus S. So, Km is basically the Michaelis-Menten constant, which is the substrate concentration when the velocity is half maximum. So, we can see over here, if we see the plot, wherever the half maximum velocity has been reached, that particular substrate concentration is called as the Km value. Many a times, generally it is said that the lower the numerical value of the Km, the tighter the substrate binding occurs over there. So, Km value gives us a identity that what will the measure of the affinity of the enzyme for the substrate for that particular reaction. Now, over here I have shown some of the very important examples where of enzymes and what are the Km value and what are the substrates which they prefer. So, we can see over here, if we see the example pyruvate carboxylase over there, the pyruvate having a Km value of 400, bicarbonate is having 1000, ATP is having 60. So, this particular thing is giving us an idea that how exactly the binding of these particular thing occurs to the enzymes over there. So, understanding the Km value is very important for understanding the enzyme kinetics. Now, these particular enzyme kinetic reaction or the equation was subsequently equated into a double reciprocal plot in which both the sides were reciprocate. That's why it is called as the double reciprocal plot, and it was given up by it is many times referred as the Lineweaver-Burke plot over there. So what the scientists did is they just reciprocated both the side of the Michaelis-Menten reaction. So they came up with a value which was 1 by V naught is equal to Km divided by V max S plus 1 by V max. So if we see over here, this particular equation gives something like the mathematical equation y is equal to mx plus c, where m is the slope and the c is the intercept within them. So if we plot that particular thing, we can get a Lineweaver-Burke plot over there in which we can see the intercept which is 1 by Km, and there is a intercept in the 1 by V naught or the maximum velocity which is 1 by V naught, and the slope is basically Km by V max. So by this understanding we can take up what exactly the maximum velocity will be, because in the previous Michaelis-Menten plot we were unable to calculate the exact maximum velocity, but in the Lineweaver-Burke plot we can easily calculate the maximum velocity to a precision. Now the next important thing knowing about the enzyme is the turnover number of the enzyme. So which is referred as the kcat value. So kcat is basically the constant and the cat refers to the catalytic reactions which this particular thing is doing up. So the kcat values gives us a equation that how exactly the enzymes are functioning and what are the turnover numbers of the enzyme. So this gives up how many reactions are occurring in a unit time at one active site within that particular enzyme. So by extrapolating, it comes up that the Vmax is equal to kcat Et, where Vmax is the maximum velocity, kcat is the turnover number of the enzyme, and Et is the enzyme total. So the kcat can be written as Vmax divided by Et. So by knowing the kcat value, we can tell the catalytic activity or the turnover number of the enzyme, means how many reactions this enzyme can go about in one unit time in a particular active site. So over here I have shown a table in which the turnover number per second is shown over here, how exactly the reaction is occurring. So you can see over here carbonic anhydrase, which is one of the important enzymes within the body, the turnover number is 6 lakh per second. So you can just imagine how fast this particular reaction goes about in the presence of that particular enzyme. Now there is another term which comes up which is called as the enzyme efficiency. So the enzyme efficiency is basically characterized or rewritten as kcat divided by Km. So this gives us a measure of the enzyme efficiency or the kinetic constants which are there. So the kcat by Km is equal to K1 K2 divided by K minus 1 plus K2. So all these K represents the various constants within that particular reaction process. So the various kcat by Km are given over here, which gives a catalytic efficiency of these particular reaction and how exactly these particular reactions are going about. Now we come into the next topic, which is called as the enzyme inhibition. So the enzyme inhibition plays a very important role in inhibiting enzymes. So the enzyme inhibition have been classified into two major classes. One is called as the non-covalent binding classification, the other one is called as the covalent binding classification. Now in the non-covalent binding classification, there is a reversible inhibition which is occurring. So reversible inhibition, as the name is suggesting, so the inhibitor comes and binds to the enzyme, but it can be reversed because the bonding which is taking place between this inhibitor and the enzyme is a non-covalent bonding which is occurring. So whenever there is a breakdown of that particular bond, the enzyme is free for another reaction and the inhibitor is made free. Now the next class of the inhibition is called as the covalent binding. So in the covalent binding, what happens is there is a irreversible inhibition. Many a times it is referred as the irreversible inhibition over there, and in the irreversible inhibition, it has been classified into three major types. One is the group specific reagent, the other one is called as the reactive substrate analogs, and the third one is called as the suicide inhibitor. And as the name is suggesting, the inhibition is an irreversible kind of reaction. So the bonding between the inhibitor and the enzyme is a non-covalent, so it cannot be made free and the enzyme becomes dead over there by the binding of the inhibitor over there. Now in the non-covalent binding, which is referred as a reversible inhibition, it has been classified into three major types. One is called as the competitive inhibition, the other one is called as the uncompetitive inhibition, and the third one is called as non-competitive inhibition. Now in competitive inhibition, in the first one, what happens is the inhibitor binds only to the free enzyme. So whenever the enzyme is present, the inhibitor will compete with the substrate and bind to the enzyme. So there is a structural resemblance between the inhibitor and the substrate over there, and as soon as both compete, the inhibitor outrights that substrate and binds to the enzyme active site, and it blocks the enzyme in a reversible way. The uncompetitive inhibitor, the inhibitor binds only to the enzyme substrate complex over there. Only when the enzyme substrate complex is formed, the inhibitor will come and bind to it, and then only the reaction will be stopped over there. And thirdly, the important class is called as the non-competitive inhibition, which is many times referred as the mixed inhibition model systems also. So the inhibitor can bind both either to the free enzyme or to the enzyme substrate complex also. So these are the three major types of the non-covalent binding which is there. Now we come into each of them one by one. The first one is the reversible inhibition, which is shown over here. Now the reversible inhibition, the first type is called as the competitive inhibition. So in the competitive inhibition, as I told you, the inhibitor and the substrate competes for binding to the enzyme active site. So at one of the image you are seeing that to the enzyme the substrate is bound, while in the other image you can see the competitive inhibitor has been bound. So to the enzyme, when the substrate binds, it forms the enzyme substrate complex and the reaction moves like the enzyme plus the product. But as soon as the inhibitor comes and binds over there, the reaction cannot move further because it has blocked the enzyme active site and the substrate cannot bind to it, and finally there is an inhibition of the reaction which takes place, and the enzyme inhibitor complex blocks the reaction and there is no further reaction which takes place over there. Now if we see the kinetics of this particular inhibition, which is the competitive inhibition, it can be seen that both in the Michaelis-Menten plot and the Lineweaver-Burke plot, the Km value was raised and the Vmax value remained the same. So we can see over here the green line in the Michaelis-Menten and the red line in the Lineweaver-Burke plot. It is showing the reaction in the presence of the inhibitor, so we can easily make out the Km value was raised and the Vmax remained the same over there, and the equation for this particular reaction, which is the competitive inhibition, becomes like this in the case of the Lineweaver-Burke plot equation. Now one of the main important and interesting example of the competitive inhibition is the methotrexate drug. So methotrexate is a very important drug which is being used in certain types of cancers as well as treating many of the severe diseases like the rheumatoid arthritis, cirrhosis, and like that one. Now it was discovered by a Indian born scientist. His name was Dr. Subba Rao, who discovered this methotrexate. And what he did is he used this particular concept of competitive inhibition in discovering this particular drug. Now what he found that the methotrexate, it competitively inhibits an enzyme which is called as the dihydrofolate reductase. So DHFR is a very important enzyme which participates in the synthesis of the tetrahydrofolate reaction within the body. Now folates are an essential part of the purine and the pyrimidine base synthesis. So you can understand, if the tetrahydrofolate synthesis don't occur, there won't be any synthesis of the purine and the pyrimidine base which will be formed, and subsequently the cell will die off. Now what he used is, in the cancer cells he found that these reactions were occurring in a very high spaced up rate. So what he did is he used a competitive inhibitor which was methotrexate. So the methotrexate was having a structure which was resembling the structure of the dihydrofolate. So both of them competed to the binding to the enzyme which is DHFR, dihydrofolate reductase over there, and once the inhibitor got bound to the enzyme, the enzymatic reaction cannot go further and there won't be any reaction for the synthesis of the folate over there, and the process of the cell will die automatically. So that's how the competitive inhibition mechanism was used in the development of the methotrexate, which is one of the important drugs nowadays. Now the next one is called as the reversible inhibition, which is the uncompetitive inhibition. Now in the reversible inhibition, if we see over here, the inhibitor binds to the enzyme, it binds to the enzyme, and as it binds to the enzyme, it can only bind whenever there is an enzyme substrate complex which is there. So the inhibitor can bind to the enzyme only when the enzyme substrate complex is there. So it forms a ESI complex, which is the enzyme substrate inhibitor complex, and once the ESI complex is formed, the reaction cannot go further. Now if we see the enzyme kinetics over here, the Michaelis-Menten plot as well as the Lineweaver-Burke plot over there, we can see that the Km and the Vmax value were lowered in both of the cases. So in the uncompetitive inhibition or the reversible inhibition, what happened is the Km and the Vmax value were lowered over there. So we can see the Michaelis-Menten plot as well as the Lineweaver-Burke plot. So both of them were lowered over there, and the equation becomes something like that, which is shown in this particular slide. Now the third important type of the inhibition system is called as the non-competitive inhibition. Now as the name is suggesting, non-competitive inhibition, so what happens is the inhibitor can bind to either the free enzyme also, or it can bind to the enzyme substrate complex also, forming the ESI. So as it binds to the free enzyme, the reaction cannot go further and it will be blocked over there. And finally, if the ESI complex is also formed, it will be blocked over there. Now if we see the example over here, the example which shows over here is an example of the non-competitive inhibition. So both the plots of the Michaelis-Menten as well as the Lineweaver-Burke plot, which is shown over here. So we can see over here the Km value remains the same. So the Km value remains the same. The Vmax is lowered in the presence of the non-competitive inhibitors. So we can see over here the green line in the case of the Michaelis-Menten plot and the red line in the case of the Lineweaver-Burke plot, how the Km value remained the same while the Vmax value was lowered in the case of the non-competitive inhibition over there. So and the other dotted line which is shown is where there is no presence of inhibitor, so no inhibitor is present, which we discussed in the previous slides about the enzyme kinetics. Now we come into the next segment, which is called as the irreversible inhibition. So till now we discussed about the reversible inhibition, in which it was classified into various three categories, okay. And in the reversible inhibition, as the inhibitor bound to the enzyme or the enzyme substrate complex, when the inhibition is done, the inhibitor can be released from the enzyme, and now the enzyme is free to go for another round of reaction. So the enzyme is not destroyed or it is not distorted in the case of the reversible inhibition. But in the case of irreversible inhibition, there are certain reaction which occurs. What it does is it changes the conformation of the enzyme active site or the enzyme, so that the reaction cannot go further within this particular thing. in this one the first type is called as the group specific reagents. So this group specific reagents are specific for the R groups which is present in the amino acids. So if we see over here, one of the group specific reagent which is shown as the DIPF, which is shown over here, and the enzyme is shown. So we can see over here there is a hydroxyl group which is present over there. So this hydroxyl group binds to the DIPF reagent over there. So the group specific, as it binds, this becomes a irreversible inhibition, so it doesn't get released from the enzyme active site. So the enzyme active site is now occupied throughout, and no further reaction can occur in that particular enzyme active site, and the enzyme eventually dies off. Now this is a example which is shown. So we can see over here the example how the enzyme was inactivated over here. So the DIPF, which is the diisopropyl phosphofluoridate, which is shown over here, it inactivates a enzyme which is called as the acetylcholinesterase. So acetylcholinesterase is a very important enzymatic reaction which is present within the synaptic transmission. So within the synaptic transmission, so you can understand if this particular enzyme is blocked, so there won't be any synaptic transmission which can occur. So DIPF belongs to a group of compounds which are many times referred as the nerve agents, because they directly inhibit the synaptic transmission by irreversibly binding to that enzyme and inhibiting that particular enzyme. So this acts as a group reagent. Now the next particular irreversible inhibition comes as the reactive substrate analogues. So over here the affinity labels are there. So these are basically the molecules that are structurally similar to the enzyme, but they modify the enzyme active site by binding with the help of the covalent bonds over there. So they are referred as the reactive substrate analogues over there. So over here if we see example which is the TPCK, which is shown over here, we can see that the TPCK binds to the chymotrypsin enzyme over there. So what it does is it binds to that particular enzyme active site, thereby the substrate cannot bind to that particular thing, and it irreversibly inhibits the reaction of that particular enzyme. And the last category is called as the suicide inhibitors under the irreversible inhibition. So as the name is suggesting, they are suiciding themselves to block or stop the catalytic mechanism reaction. So what happens is, initially the rate of reaction catalytic mechanism occurs in a normal processed way, but the product or the intermediate which is formed, it is a chemically reactive intermediate. What it does is it comes and blocks the enzymes through a covalent modification. So the inhibitor binds to the enzyme as a normal substrate, and it initially initiates the reaction processing the normal catalytic mechanism, and finally the chemically reactive intermediate which is formed, it goes and inactivates the enzyme by binding and acting through a covalent modification over there. Now if we see over here, the example which is shown over here is an example of the aspirin over there. So we can see over here the COX enzyme which is there. So there is a amino acid hydroxyl group which is present over there. The aspirin which comes and it binds to that particular enzyme active site, thereby producing the reaction, and subsequently the reactive molecule which is formed or the intermediate which formed, it inactivates that particular reaction and it blocks that particular reaction process over there. So the aspirin acts by covalently modifying the enzyme which is the cyclooxygenase over there, the COX enzyme which is shown over here, and reducing the synthesis of the inflammatory signal, thereby reducing the other sensations which are in a signal transduction manner which is occurring over there. Okay, so these are the various reaction steps which were shown over here. So in this part of the lecture, I discussed about the enzyme kinetics, what is the Michaelis-Menten reaction, then the Lineweaver-Burke plot I discussed about how exactly the enzyme follows these kinetics and what are the various steps are present, then the classification of the enzyme, and finally the enzyme inhibition, where it was broadly classified into two major classes. One is the reversible and the other is the irreversible. And under the reversible we discussed about the various class, the competitive, non-competitive class, and under the irreversible we discussed about various reagents, how they block the enzyme. So with this note I come to the end of this part of the lecture. So hope to see you in the next part of the lecture. We will be continuing with another set of topic on the biochemistry. Thank you very much for your time. Thank you very much. Thank you.